Hello listeners, Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, I have a few announcements to make. We are working on a new program geared towards transitioning from academia to industry. Powered by Dr. GPCR, our new program is called Life Career Job. The goal is to support you in your efforts to find your next job in industry, plan out your career trajectory, create a life and support the lifestyle that works for you. If you'd like to be notified when this program becomes available, fill out our short survey at lifecareerjob.com. This new ecosystem will also include a new podcast that will be launching in September 2021. Our guests will be scientists, recruiters, and hiring managers, and everything in between. We want to help PhDs find their ideal career path and inspire you with real-life stories. The second edition of the Dr. GPCR Summit will be held from September 13 to 19, 2021. We are planning a combination of live talks, pre-recorded talks, and live workshops. We have a great lineup of live talk speakers, including Dr. Brian Roth, Dr. Patrick Sexton, and Dr. Jürgen Weiss. Also, we are happy to announce that we'll have a live Q&A session with Dr. Bob Lefkowitz and Dr. Randy Hall to talk about GPCRs, science, and Dr. Lefkowitz's memoir. Visit drgpcr.com/summit today to see the full program. We're also launching our GPCR consulting services. You'll soon be able to find the profiles of our carefully selected consultants on our website at drgpcr.com/consulting. You'll be able to find the help you need for your company. Stay tuned. Are you subscribed to our YouTube channel? If not, please subscribe today. It's not only a great way to catch up on our recorded events, such as the Dr. GPCR Virtual Cafe, but it's also a great way to support Dr. GPCR. And now, let's dive into this episode. Hello everyone, this is Yamina from Dr. GPCR. Welcome to our second live Dr. GPCR podcast recording. Today I have the pleasure of having with me Dr. Ines Liebscher and Dr. Simone Prömel. It is a live recording, so we have multiple guests joining us. A couple of housekeeping items before we start talking to our guests. Please make sure that your microphone is muted at all times. I opened a chat box. If you have questions for our guests as we go through our podcast episode, please make sure that you put them in the chat box. We are about 8 participants total, so 5 guests. Just make sure that you put that question in the chat box, and I'm happy to read them to our guests and they can answer your questions. So thank you all for being here, and let's just give it a go. Hold on, before we do that, I'm bringing up the chat box and making sure that everyone gets access to asking the questions. All right, so I think we should be good to go. And before we go, I just wanted to show off my brand new Dr. GPCR mug, which is not for sale. It's a gift that I got from my husband for Mother's Day, but everyone has been complimenting me on it, and I'm pretty happy that I can finally show off a nice cup. All right, so let's do this. Good afternoon, Ines and Simone. I'm so happy to have you on. Thank you so much for coming today.
It's great being here. Thanks for the invitation.
Thank you very much for the invitation.
Fantastic. So this afternoon, we're going to talk about both of you, your careers as early career scientists, but also about adhesion GPCRs. So let's start off with some introductions. And I am going to pick Simone. You're on the left of my on my screen. So please introduce yourself to the audience.
Well, my name is Simone. I'm a biochemist by training. And currently I'm based in Düsseldorf, where I just started in February as a professor for cell biology. And naturally, I'm working on adhesion GPCRs. And that's my passion. And that's basically what my lab is working on.
Thank you. Ines, your turn.
Well, I'm a medical doctor by training, but after I finished all the exams, I felt that I really just want to keep on doing science, which I already started in due course of those studies with this MD-PhD program, and I just went to finish this, started my group, went from there. Right now, I'm a professor for biochemistry at the medical faculty here in Leipzig, with basically a focus on signal transduction. And it's been ever since I started postdoc, it's been adhesion GPCRs for my research. But before that was just plain other GPCRs. So basically all kind of stays the same.
That's fantastic. So we're definitely staying in the GPCR family today. Before we move on to talking about GPCRs, I'd love to ask both of you the question about how did you end up in science and how did you end up working on GPCRs? And Ines, it's your turn this time.
Well, I don't know. I kind of always wanted to study medicine, but I kind of also always wanted to do science. I know it sounds really terrible, but I knew that from the beginning. I mean, with my family history, I'm kind of prone for that. My brother was sick, and you see that, and you don't really understand what's going on. So that was, I guess, a big driving force for me. And I always thought I might change my mind, but in the end, I just went to study medicine and do the research, and I never regretted it. And the topic of GPCRs, I mean, that was one that I didn't really choose. I was basically looking around, and as I was taking my first state exam, I was at that point already registered with some scholarship network in Germany, and they had something they called research meetings networks. And I had to read some papers at that point, and I didn't have access. So after my exam, I asked basically one of the professors if he could help me get access to these papers. And from there on, basically, we were talking about these papers, and then he said, hey, you could do your thesis with me. And well, let's put it like that. I stayed on for many, many years.
So just from asking for papers, you ended up doing your studies there, which is phenomenal.
Yeah.
Simone.
Well, when I was younger, I always wanted to become an historian. And that was basically what I always wanted to do. I wanted to become a historian. I wanted to look through papers and books and find out, basically do research on history. And well, then I had a friend who studied history, and she was a bit older than me, and then she got unemployed, and she was like, well, don't do it. And I was like, well, okay, what else could you do? And then, well, I figured I was always into nature, and that was basically my second choice. And I studied biochemistry, and from the beginning, it hooked me basically. So I did biochemistry as an undergrad, and I figured out quite quickly that I wanted to do science, really. So I moved on to basically do a PhD, and well, as a biochemist. And well, that was when I first came across adhesion GPCR. So they basically chose me, and I didn't choose them, because I really liked the topic where I did my PhD. They did cell biology, they did developmental biology, and they did adhesion GPCRs, and that's how I moved into adhesion GPCRs. And basically ever since, I never let go. And I moved to Leipzig, where I met Ines, to basically, after my PhD, build up my own group. And I developed into adhesion GPCRs, and yeah, I never left.
So you kind of both grow into the adhesion GPCR family. How interesting, a historian. I think you're the first guest ever who says a historian. I've had musicians, I've had I think even stand-up comics, but I'm not sure about that. I think that reminds me of someone I know who may or may have not been on the podcast, but a historian. What an interesting way to end up as a scientist.
Well, maybe I do it when I'm retired, become a historian.
Who knows? I think you're still using a lot of those skills. You know, the interest in the past, the interest in what has been discovered before, but also it gives you an idea on where you want to take your research ahead. So, I would just want to take a little step back. So you both ended up in the GPCR field during your PhD. You were exposed to adhesion GPCRs during your PhDs. And what led you to continue on? What did you find fascinating about these receptors that led you to continue on? Simone.
When I started my PhD, there was not much known about adhesion GPCRs. As I mentioned, I moved to developmental biology. That was basically my angle at that time. So it was physiology at first. And then we discovered that some of these receptors do have a function there. And then we went through the literature, and then we figured out that there wasn't really much known about that, neither about signaling nor about basically function. And that is why I got interested in these molecules, because when you basically study biology, you come across GPCRs, and there's a lot known, and there are a lot of them. And then you all of a sudden figure out, hang on, there are still quite some of them where there's not much known about, and how could that happen? And that is why I got really interested in them. And when you have a closer look at these guys, I mean, they're huge, and they have a lot of different function, signaling wise and functionally. And so there's a lot to do. And every day, basically, you can move into different direction. And that is what I love about them, so that they always surprise me. And that's why I wanted to stay on.
That's fantastic. And this period was in the mid 90s, mid 2000s?
No, that was around mid 2000s, basically.
Ines?
Well, I have to be honest here. I stayed on just because I really didn't feel like giving up. It was it was a really insane start to this. So I finished med school, and then we were thinking, okay, what could be a good postdoc study, or for the PhD, basically at that point. And then Thorsten, my supervisor, he was like, oh, I have this really cool thing, these adhesions. The clinicians were coming up here. They found this receptor, and I think it's a really cool field to get into. You just have to, there's nothing known. You just have to clone one and do a little bit of signal transduction. That's basically going to be the paper. Super easy. Well, let's put it like that. It took me a year to clone the first adhesion GPCR, because at that point, I mean, there was really, you couldn't even buy them. They were too big to synthesize them, at least for me as starting up. I didn't have the money to do that. So basically, we were getting these receptors from messenger RNA. So the mouse ones were still okay. We had access to that. But basically, all the human adhesion GPCRs that I deal with, they're from my blood, basically, because I took my own blood. And then I was sitting here at night with friend trying to isolate the leukocytes and then prepping the RNA out of it, and it used to be such a mess. And I just felt like, okay, you cannot stop doing this. And then basically, once you got one, you got a lot more. And then we figured out that they really do a lot of cool things. And every single time you answer a question, you end up having 10 more. And for some reason, I just can't let it go, even though in between, I mean, seriously, there were frustrating times, right? And I was thinking, you should be wise. My friends from studies, they're like, why don't you go back to clinics? I mean, really, seriously, the salary, the contracts are limited. What are you doing? I was like, I know there's this fallback option, but I really, really just want to get this one more question, this one more answer to that question. And here we are, right? Still doing it.
That's phenomenal, from your own blood. I love that. I really love that. And I guess one of the issues must have been that at that time, it wasn't as easy as it is today to say, well, I have a sequence. And if you have a sequence, you get it synthesized and you're good to go. Because sometimes you didn't even know what the sequence was, or synthesis was not an option.
Well, actually, the problem is that a lot of those sequences that you could find in the databases is that you just found some artificial transcripts, because they were pulling them up or finding them. And these adhesions are really huge. And we just really started discovering several years back, I mean, not me, but other groups, what is this huge repertoire of splice variants and so on. So it was really, you were trying to fish for something, you design your primers, and then in the end you get, like PCR bands that totally do not match what did you expect at that point.
Oh goodness, I can't wait for us to talk about your favorite GPCRs, your favorite adhesion GPCRs. But before we do that, I want to ask both of you, how did you meet? When and how did you meet? Any of you?
It sounds like it.
Well, we actually talked about that prior to this episode, where we were reminiscing about how we met. We actually met in 2011, I think it was, at a GPCR meeting in France. So I just had finished my PhD, and I was basically I had an intermediate postdoc where I did neuroimmunology because I went through that phase afterwards where I thought, well, now is a chance of maybe looking into something completely different and get some more technical experience before moving back to adhesion GPCRs. But I gave a talk at that meeting, and that's where we basically met.
Yeah.
And we didn't know that we were ending up in the same place because Ines' boss was there as well. And we got into talking, and he was like, hey, I was in the UK back then. And he said, well, if you ever want to move back to Germany, then give me a ring and we can arrange something. And as Thorsten was interested in adhesion GPCR a lot back then, it somehow matched up a year later that I moved to Leipzig, and Ines was already there. So that's where we basically started our research together, but we actually met a year in advance.
Yeah, but I remember that meeting because I was really just starting out with these, and these adhesions, they all have these numbers. It's all numbers. I mean, now there's a new nomenclature, but that somehow doesn't really make it much easier after I finally got used to all those numbers. And I was talking to Simone, and she came to my poster, and she's like, so is this one doing something like this other receptor is doing? And she started giving me all those numbers, and I was like, you know what? I know this one guy that's right here in the title of that poster. But apart from that, I know they're there, but I don't even know what you're talking about.
It was actually a quite nice conversation. So she never came across that.
And that was 10 years ago when you think of it. It's 2011 to 2021.
Yeah.
Oh my God, that sounds terrible.
I don't know. I think it all depends. It was, as I was mentioning, my brand new mug, my previous mug, which I try to hide whenever I'm recording a podcast episode because it has flowers and, cows on it. That mug is 25 years old, which I think it's even more horrible than you knowing each other for 10 years. All right. So let's dive into adhesion receptors. Let's say I'm a graduate student. I know nothing about adhesion receptors. What would you like to tell me about them in a minute each? Ines.
Well, they're super exciting. They're a pain in the ass, but they're great. I mean, in many aspects, they are great in size, and they're great in secrets, and they're great in discoveries.
How big are these receptors, and how many members are in the adhesion family receptors that are known, at least?
Well, we have 33 in the human genome. And the biggest one, what was that? 6,800 amino acids. I always like to give those numbers when we start going into the talks. It's like, the C-terminus of an adhesion GPCR can be up to 400 or 500 amino acids. And just an average other GPCR is about 400 amino acids of size. Right? So you can fit a whole rhodopsin-like GPCR into the C-terminus of an adhesion GPCR.
Wow. Oops, hold on. We have a new guest coming, and I'm just going to mute. That's phenomenal. That's phenomenal. And do we know, so you said 33 adhesion GPCRs. Are there any classes of these GPCRs that can be, divided can these be divided in subclasses? What do they do exactly? And we're really into the one-on-one of adhesion GPCRs before we move into more trickier questions. Simone.
33, as Ines already mentioned, can be subclassified, as you want to say. There's been a long debate, and I think it's not finished yet about that. At the moment, we have 8 major classes plus one. Based on phylogenetic analyses. So the first nomenclature, first of all, they were basically classified as being class B GPCRs because together with the secretin receptors. At the beginning, they were like in the late 90s, they were always termed long N-terminus 7 transmembrane receptors. So LNB7TM receptors. So when you read old papers, that's what you basically come across. And that's what they are called, or where they were called. And people started thinking of like, what the hell is that a name that no one can remember? And that's really boring. So people looked into phylogenetic aspects, then it became pretty much clear that they're not really like secretin receptors and not really like class B receptors. So then the GRAS classification came up and they got their own class, or they became their own class. And then the subdividing basically took place. So we have these groups where you can, based on the 70M similarities, you can group them. But there's still a huge debate on whether this is the right way of doing it or whether you should basically group them differently. And that was also the time where the name adhesion GPCR was coined, because people found out that they have a lot of domains where they didn't know about anything in the context of adhesion GPCRs. They were just there, and people thought like, well, they look like adhesion domains. They do adhesion in other contexts, so let's just call them adhesion GPCRs. And that's where we swapped from long N-terminus 70M receptors to adhesion GPCRs.
Which let's be frank, it's much easier to remember and to write
catchy
as well.
And later on, people found out that they really do mediate adhesion shows. So it is true, at least to that extent.
That's phenomenal. Do you each have a favorite adhesion receptor?
I think
Ines.
we all do. I mean, obviously, the one that I got after 1 year of cloning, GPR126, that was the one where we also got the first signal transduction. Basically, it's the workhorse for anything new that we came up with. Ligand activation, mechanoactivation, small molecule compounds. So the 126 is just a favorite for me. But we also work a lot with GPR133. Which is a bit of a different one. But to be frank, basically, a lot of the collaborations just end up people, saying, I have this adhesion GPCR. Can you tell me what is it doing? What is it doing? So we're dealing with all of them, but my favorite pet is still GPR126.
The first one you were able to clone from your own cells, which is phenomenal. Which I think it should have a favorite, a sweet spot in your heart.
That's why you need medical doctors to do science. They know how to get access to human material.
Yes.
I tried corpses, by the way. We had the anatomy, and I was running over there trying to get some samples, but the bodies were never fresh enough.
Oh goodness. Spoken like a true physician. How about you, Simone?
My favorite receptors is similar to Ines. That's the first one I encountered, and has a place in my heart forever. I think these are actually in the mouse. There are 3 of them, or in humans there are 3 of them. There's electrophiles belonging to the older ones, phylogenetically speaking, the older ones. And they are present in vertebrates and in invertebrates. And I'm a big fan of model organisms. Give me any, I like them. So that is why I really like electrophiles, because you can find them basically everywhere. And they are doing a lot of funny and cool things. And obviously they were back then involved in developmental processes. And that's when I got interested in them. But yeah, I like what they're doing, so I follow them. They don't follow me, I follow them. So they are the ones. I have others. We have others now. We're working on seltzers as well. We're working on GPR 110. But if you ask me if I had to pick one, it's basically electrophiles.
That's phenomenal. So and this is a question for both of you, and we can take turns. You can take turns to answer that. Since you've identified a favorite GPCR that you like, let's take a little bit of time and talk about the current status of research of your favorite GPCR. So Ines, GPR 126, you've cloned it. You've basically bled to clone it.
Yeah.
What does it eat in the winter?
Well, I mean, it's actually one of those nice classic G protein signaling ones. There's other adhesions that are not doing us the favor of doing that easy readout. Yeah. So that was nice. But what's still picking us up every day is that we keep on finding, well, actually not us. We have great collaboration partners that keep on finding interaction partners. We call those ligands interaction partners. Because the real agonist for adhesion GPCRs is basically a tethid agonist. So we are really trying to figure out how do all those different ligands mediate a signal. And what's really cool is that we found that depending on what type of ligand that is, if it's a certain type of collagen, it can be a laminin, they all have a different mode of activation. They can actually induce signaling bias, as far as we know. And what's cooking up right now is that we were actually determining and quantifying the forces that you need to induce activation with those different ligands. To kind of, because that question always came up, we were talking about mechanoactivation. We were really doing some nasty experiments at the beginning, just detaching cells, shaking them, vibrating them. And everyone was always, yeah, that's great. But, what's the physiological meaning, right? The holy grail. So we did that, and we actually got some really, really cool results, which we're hoping to bioarchive in the next weeks. So this is really cool about this one. I mean, there's also a lot of nice physiology about this receptor. It's just that this is what other people do, because that is, if you knock it out completely, then the animals kind of die. So you have to deal with different mutants and variants. And there's some really cool stuff on myelination, scoliosis, and so on and so on. So that is a part that we don't touch. We just really do the activation, and are really happy to work with the people that do the physiology together with them and see what could be causing these phenotypes.
That's phenomenal. So you mentioned signaling bias for GPR. I want to say GPCR, GPR 126. What's the G protein coupling look like? What mediates that signaling bias? Can you please tell us a little bit more about that?
Yeah, so actually 126, if you stimulate it with basically a synthetic peptide that is derived from the tethered agonist, you get GS, GI, and GQ coupling, and basically whatever you can think of downstream effectors. We do not really look at 12, 13, but since we know there's SRE and SRF, there's probably also that there. So when you have a full activation capacity, this receptor basically takes anything. It's dating all of them. So what's really interesting then is that always was the question, how does that make sense, right? And we found that the first indication we had is that when you look at different activity states, so if you just express the receptor in its full length state, you only get the GS signal. But if you then start, inducing some constitutive activation, there's quite a classic thing known in the field. If you chop off this N-terminal fragment, then they get really constitutively active. Then you get GS and GI, and only when you then keep on adding that peptide, then you also get all the others. And just basically by chance, by cooperating with those people that found those interaction partners and asking us to do the pharmacology with that, we found that it really matters which type of basically extracellular matrix molecule you add. And they differ in a way. I mean, of course, a collagen is not a collagen, so they differ in their own molecular characteristics, but also they differ at the place, at the N-terminus where they bind. And we haven't really gotten all the way down to showing that specifically, but we basically from the first experiments had the idea that it could be something like physics or maybe, interconnection between N-termini. But in the end, it's some of them induce the signal by basically just lying on the cell, while others need some additional pull to do the activation. And that is something that we did, as I said, with these really nasty experiments of, putting shakers into incubators and this kind of stuff. But now we actually have the forces with it, and we know for which types do we need the pulling, which types need the pressing on. And that's actually really interesting because it seems that even they just need to interact with the N-terminus. And by that, they apply this type of pressure on the membrane. We're not sure if it's the receptor or the membrane that's then actually doing the activation cascade. So there's still so many more question marks as we go along, but I feel like we're shedding more light onto this.
That's phenomenal. But I just find it phenomenal because, first of all, they're huge receptors, which again, as soon as you have a very long C terminus, then the question is, does it get phosphorylated? What kind of interaction can it do? Can it have with other signaling proteins, but also with the long N terminus that can serve as a nice way to, interact with the ligand and kind of place it in kind of in, I want to say in a Lego type of manner. You want to make sure that you're getting into there. I think it's really phenomenal. Let me ask you a follow-up question about that. What is the physiology that is known about GPR 126?
Well, the most or the best established one is the myelination deficit that you get. And actually, that is the phenotype where we can really nicely match the different signaling cascades. So you need GPR 126 to move a Schwann cell into the differentiating states where they actually start wrapping the neuron. And to induce that, at least that's what Schwann cell people say. It's really not my field of expertise. They said you always need a burst of cyclic AMP. And we found that this actually matches very nicely with the expression of laminin 211, which starts building the basal membrane at the beginning. And it's that laminin 211 that when it just sits on the receptor, it keeps it in an inactive state. So you're not activating GI, but you're keeping GS inactive. And then as the laminin 211 starts polymerizing, it's somehow exerting a force on the receptor. And in the lab, we were basically mimicking that force by applying this vibrational additional force onto it. And in vivo, they could basically get a good indication that it's got to be this polymerization, because when you try and rescue that phenotype in zebrafish, you can do that. So you have a hypomorph mutant of the receptor, and the zebrafish gets that phenotype. And when you then overexpress the receptor or laminin 211, you rescue that phenotype. So it's not completely gone, but much, much better. But then when they introduced the laminin 211 that cannot polymerize, that phenotype could not be rescued. And this was really, really cool stuff done in the Kelly Monk lab, actually. And that fit so nice. There was such a nice story how we always played it back and forth, the stuff that we found in vitro. And then they could look this specifically up in the zebrafish. That was really great. So we get a good hint there. But there also, GPR 126 is also doing heart development. There's quite a lot of stuff known there. It's shown for bone development and tendons, as far as I know. So they're ubiquitously expressed almost.
I find it fascinating that it can interact with different G proteins depending on whether you're adding some force to it and depending on the ligand. I think it's just it must be very tightly controlled in order to exert its function at the right time, at the right place, within the right conditions, which I think it's
exactly. It's sensing its environment.
It's awesome. I think it's just phenomenal. I my favorites are chemokine receptors, and just the fact that there are more chemokines than receptors. And for a long time people thought these relationships were promiscuous, which by now we know they're not. I think it's just beautiful. But having the ability to control by mechanosensing or mechanoactivation of a receptor, I think it's just phenomenal. So Simone, well, what about your favorite receptors?
Well, when I started off, electrophiles were one of the receptors in the field of adhesion GPCRs where there was at least something known. So I didn't start off from a completely blank spot. However, they are called electrophiles because they bind, or the toxin of the black widow spider, the latrotoxin, binds to it. So that was known at that time when I started. So we stumbled across them because they had to function in cell polarity. So I came from the physiology side when I started my PhD, and we were interested in cell division and how a cell basically finds its neighbors and how a cell knows where it is and what it is and when to divide and what to become. And electrophiles were basically involved in that, and that what we found, and that is where I started off looking into them. And then the question arose, hang on, how do they really do that? And there was not much known about signal transduction. So back then, it wasn't even shown that adhesion GPCRs are actually GPCRs. That only came later. So we got interested, or I got interested in understanding how they mediate that basically physiology function. And so we looked into, first we looked into the large, extraordinary long N-termini, because we thought, well, it might be quite useful to understand what binds to it and how, they sense their environment, as Ines said. And that's where when we find out what signals they mediate, they couple to GS in that sense. They can also couple to other things. But what we found, and what still basically interests me, is that this large extraordinary N-terminus cannot only basically act as a tethered agonist for activating the receptor. It can do so much more. It can do adhesion, which other people found. And it also can mediate functions on different cells. It can also act as a ligand itself. And this is what makes adhesion GPCRs even more complex than they are. And this is what we followed up. So we found that they basically do a function in cis into the cell, like a classic GPCR mediated function. And they also act as a ligand. And what we try to understand at the moment is, how does the large N-terminus integrate all these kinds of signals? So how does the N-terminus basically mediate a signal into the cell, and when does it do it? When does it decide to act as a ligand for a different cell, and when does it mediate adhesion? And we know that electrophiles can do all three of that in different contexts. So, and this is something which really we try to delineate at the moment, and which is really cool, because what we also found is that it was something based on a publication by Thorsten Schöneberg, that they found that adhesion GPCRs have a lot of splice variants. So we thought, well, the N-terminus might mediate, or basically might be composed of different modules which are brought about by splice variants. And we looked into that, and then we found that not only the N-terminus is basically modulated by different splice variants, but also the C-terminus, and that also mediates different signals. So they are far more complex than they seem to be, and this is what we try to delineate in different functions. So we try to understand, when do they signal in cis, when do they signal in trans, and in which contexts. So that is basically what interests us. And at the moment, we're basically looking not only in cell polarity in an embryo, but also in pancreatic development and in metabolism, where you also have a lot of polarity going on, and where basically we can delineate those two functions, the cis and the trans function of these receptors, and to understand which ligands bind to them. Are they different ligands? Are they the same ligands? And so this is really tricky to delineate.
I find it fascinating that, within the same family of receptors, you can actually clearly distinguish between the function of different splice variants. And I think that's just phenomenal, because not only you have multiple members of the same family, but you have splice variants, which again increases that diversity. How does the pharmacological landscape of electrophilins look like?
Well, there's not that much known about that. So we have a couple of ligands. Actually, we have electrophilins are, that's basically grown historically. They have basically, what's known is that they have a function in the synapse, so in the brain basically. They were discovered as being a toxin binding receptor. In the basically nervous system. So that is why people looked into that. And there's a lot known about how they function on a synapse, what they bind, but it's been not that much clarified what the pharmacology behind that is. So we found we tried to identify the G protein coupling, all three of them. So we have electrophilin 1, 2, and 3. Interestingly, we know that electrophilin 1 couples to GS, and electrophilin 3 couples to GI. And in pancreatic beta cells, they're basically opposing each other. So they're both in pancreatic beta cells, they're opposing each other. And depending on which signal basically is stronger, as to speak, you get a modulation of insulin secretion. So they both modulate in an opposing way insulin secretion by basically coupling to different G proteins. And this is something which we found quite interesting. And so that is basically what is known about the pharmacology. Other people think that electrophiline 2 couples to GQ. But it's not that we have basically touched the bottom of that yet. And what is also not known is none of the ligands or interaction partners which have been identified has been clearly shown to really activate the receptors. So there is basically more to it than to like binding one ligand, or basically maybe there's mechanical force involved. We don't know that yet.
I find this really, really interesting and really intriguing that that they exist, that they do things, because we can experience it, we can measure it, but we don't know exactly what the molecular mechanism of their activation. And I feel like there is also a need to better understand their expression patterns and what they do in different tissues. And it seems to me that the field is pretty exciting, but challenging at the same time. So, which leads me to my next question. What are some of the challenges that you think should be addressed to better understand the function of adhesion GPCRs, or of your favorite receptors in general? Any of Ines?
Well, what we do need, and which has luckily been picking up lately, is we need more structures. Because we did have quite a few nice structures of the N-termini on their own, but they somehow did not really allow that what we see in the pharmacology. So the TETH agonist in these structures is really tightly bound there. And it seems so tricky to think how this could be released. And I mean, there's now progress there trying to do dynamic simulations, and they identified gaps there. And now we're basically looking at ways of how could the TETH agonist interact with its binding pocket, whatever that might be. Right? This is also picking up that field. See a lot of bioarchives now pointing into ECL2. What we would really love to see is a full-length structure, and we got really excited. There's a cryo-EM structure out there, but unfortunately, the N-terminus could not be displayed clearly enough. So, but at least the good thing is we have now a 7-transmembrane domain structure. So we take it bit by bit. But in the end, what would really help solving a lot of these issues that we have here is the really full-length structure. I can't wait for it. There's something more deposited, and we're just all waiting for it to be released.
I feel like you'd be refreshing the page on a regular basis.
Yeah. Bring out that paper. Tell us what is it doing and how does it look like. Give me nice pictures.
Yeah, I guess the difficulty lies because of the size of the receptor. So, but perhaps purifying a GPCR is not an easy task. Although I feel like with the cryo-EM technologies, getting developed and getting better and better, it's easier to get a structure. Not that I have ever had to do that or I haven't done that, but having a huge receptor and purifying it and making sure that it's correctly expressed is even more challenging.
Absolutely.
Simone, what are your thoughts about the challenges that should be addressed to better understand adhesion receptor function?
Well, I think what we really need to move on into understanding them better is getting modulators out, getting molecules, let it be small molecules, let it be nanobodies, let it be antibodies against it, not only as modulators, but also as making them more visible, more present. So we don't really have antibodies against these guys. They have extraordinary N-termini. They are large. We still don't have that good antibodies, neither for marking, labeling them, nor for modulating them. And I think for really getting to the gist of all of it and to drawing apart what they can do in terms of different signals, also draw apart different variants, is having modulators and having molecules at hand, which makes us or puts us into the position of getting a bit more into what they can do and to manipulate them. And I mean, Ines already mentioned the tethered agonists. There are peptides out where you can which you can use to activate them. But it would be nice to like move forward and get different tools at hand. And I think that is one key also to move into more pharmacological and pharmaceutical ideas of these receptors to see if we can like get into drug targeting, for example.
That was my next question. So just to make sure that I understand, when you say modulators, you're thinking about tool compounds that can be used to track the function.
Among others, yes.
There's actually quite a big toolbox there. It's not just compounds. Yeah, compounds would be great, but we're all looking for ways of how you could basically use the mechanoactivation for some targeting, right? And I mean, we can do that in cell culture, but is there a way that we can tissue specifically, ideally cell type specifically, target them in a way that we can induce a specific signal? As we know, they can signal to those many G proteins, at least some of them.
Yeah.
How could we use that? And I think what we need here in terms of tools is basically we need to combine physicists doing physics.
Yes, that's the right.
That's the right one. Okay.
Yep. That's the right word.
With like in vivo, like real time readouts. That's what we need. You need to try and pull these guys or I don't know, push them, whatnot, and look at the same time at the activation rates. Mm-hmm. And that is the first thing we need to know to kind of quantify what is going on there in the cells, and then move on from there. If there's any idea how you could stimulate mechanoactivation from outside, ideally. Because the tethered peptides have a real big problem. They're not specific for the adhesion GPCR that they derive from. It's such a highly conserved region, and then they're terribly soluble. It's not great working with them. So whatever we can do to increase specificity and basically practicality to at some point feed it to a model organism, or let's even say a human being.
Yeah, but it's interesting because you were mentioning the peptides. So just to make sure, they're hard to solubilize, not easy to solubilize. Okay, hard. Okay. Yeah. But then the thought when you say mechano induction or activation, in my mind, and excuse the naivete of what I'm going to say, is that I understand that you need to get that peptide, for example, into human or into a model organism so that it reaches a target. But then the mechano activation of it, I'm having a hard time imagining it. Shaking a mouse or and it's super naive. But whenever if you need to shake the cells, or you need to vibrate the cellular environment to get that effect of that peptide or that compound, then how do you translate that into a potential application for a human disease, for a disease model? Which is kind of
the peptide. Yeah, the peptide for sure cannot do mechano. And actually, the vibrating is not so far off. I mean, we do know that there's mechanosensitive adhesion GPCRs in adipose tissue. We've been doing quite a bit of work on that, and we're looking at the ways of how they're activated. But it's actually, there's a whole field looking at mechanoactivation of adipose tissue. And there's quite a few interesting studies. It really depends what kind of force do you apply on that tissue, right? So there's this study that shows if you apply like a constant force on the adipose tissue, it was a suction force applied outside on the body, you increase adipose tissue. Well, if you have more vibrant, shaking, alternate mechanical forces, then you reduce adipose tissue. And that's not so far off from what we know from bone formation, right? You can always decide from the mesenchymal stem cell if it's going to be a bone or if it's going to be an adipocyte for these ones. And we do know in bone that you need, forces, traction to make them grow into a bone. So it's not far off, but the right point is, what is the tool to target an adhesion GPCR specifically?
Yeah.
We have a few ideas.
Share away if you can. I'm always excited to hear about new ideas. What can you share with us about those ideas?
You need something that you can modulate from outside, where we're thinking of beads that you can attach, that you can code. So yeah, I can share this now because that paper is being wrapped up and we're going to bioarchive it very soon. So then it's going to be out anyways. But actually that's the thing. I have talked about this before. It's just that also at that point I thought we're going to be really close. But the thing is when you deal these mechanoactivations, we always have the problem of reproducibility, because as it turns out, the student or postdoc, whoever's changing and dealing with these receptors, some of these adhesion GPCRs are so sensitive to these mechanical stimuli that depending on how hard do you press down your medium as you change it, how do you apply your suction, you get totally different results. And to getting this to a point where you feel secure that, okay, whatever I'm going to publish now, I can describe exact enough so that someone trying to reproduce it will end up seeing something ideally and very ideally much more to see exactly what you see. that has just really been taking so long.
I think, I think when it comes to, you mentioned suction and dealing with the cells, I find it already to be an art to take care of your cells the same way, so that each time, no matter what the passages of the cells, obviously within a certain reasonable range, or no matter which vial of cells you're using, you always want to get the same data. But I cannot only imagine the difficulty with a mechanosensing receptor where, if you don't pay attention or your hand is shaking or you're doing something off that day, then you can end up with very different results. So you mentioned beads. Can you deep dive a little bit more into that, because I'm a little bit confused about that.
So there's all kinds of magnetic beads that you can coat, and you can coat them with antibodies. And the big problem, that's really what is keeping us from doing this in model organisms, is that we barely have any really good antibodies targeting the real endogenous sequence of these receptors. But you can help yourself with artificial epitopes, just in vitro, just for the proof of principle. You can do that. But then again, it's this is a bit of fiddling around and trying to understand how many concentrations, what's the incubation time, and so on and so on. And it's been a pain. Quite a few people have been going through that, but we're getting to the point that we can describe. And I really hope that someone once this is out, is going to reproduce that and tell us that they can see the same thing, or maybe something even cooler.
That's phenomenal. But just to take a little bit of just to wrap up this part of our discussion. So they're involved so adhesion GPCRs are involved in many diseases, but there is a need for a better understanding of their pharmacology, of their physiological and pathophysiological roles, but at the same time, there is a need for modulators, and not necessarily only tool compounds, but also any type of modulators that will induce the activation of these receptors, at least modulate. It's kind of funny way of going about it, modulators that modulate the function of the receptors. A little bit more about disease setting. Are there any diseases that are known in which adhesion GPCRs play a role? And do you know if these are being investigated as drug targets?
Well, there are quite a lot, actually. So there are monogenic diseases, which have been very early on already associated with adhesion GPCRs. For example, you have the Usher syndrome, which is deafness, blindness condition, where you have a receptor called VLGR, very large G protein coupled receptor, basically, where you have mutations in that, and those people are basically deaf and blind. We have brain conditions where you have an affected receptor called GPCR56. It's called a polymicrogyria form, basically, where you have brain malformations in these patients. Recently, there was a form from vibratory urticaria, associated with one of the receptors. So there are actually quite a few of them where we have monogenic associations with them. So where a single mutation in an adhesion GPCR is causative for a certain disease setting, which is quite interesting. And these were actually diseases, except for the urticaria, which were already known very early on. So where people basically came from the disease side of things and found that we do have, mutations in there. These mutations, most of them basically, some of them basically just destroy the receptor in terms of like structure and function. So they just they are not expressed that well anymore. But not for all of the mutations that has been shown. And then obviously, and that is a quite wide field where just people start moving into recently is, data we get from GWAS studies, basically. So where we have polymorphisms in GPCR, adhesion GPCR genes, or basically close to them, where they have been, which have been associated with the etiology or with a certain risk for certain diseases. And there are actually quite a lot of them. All, first of all, to name is cancer, basically all kinds of types of cancer. Adhesion GPCRs pop up, and obviously one has to ask how true these associations are, because they have been basically pulled out of huge data sets. But there is actually a lot of potential still there, right? I mean, we have to look into that. We have to basically mine all these data. So they have not been mined properly yet for many of them. So they pop up here and there, and then you go to a conference and meet someone who says, oh, well, I'm looking into this adhesion GPCR now because it has been associated with my favorite disease. And you think, oh, wait, hang on, I have never heard about that association. So there's actually quite a lot which is to come in the future, where we probably will come up with diseases and association with diseases where adhesion GPCRs play potential minor roles, but modulatory roles.
Phenomenal. Speaking of, better understanding these receptors in disease settings and also targeting adhesion receptors, what do you think are some of the tools and initiatives that could speed up this endeavor, so better understanding these receptors in a disease context?
Well, first of all, you obviously need to understand what we're talking about. So we need to basically get people on board who are capable of looking through all the data out there, patients' data, association data, and basically get them into order, sort them, and make them accessible to scientists. So what we think basically is what we need is like a collaborative effort. So we need a lot of different expertise for that, starting off with basically bioinformaticians to get these data mined, and then obviously we need clinicians to can feed into that endeavor. So what it basically needs is huge network to get people from different fields together, not only the people who work on adhesion GPCRs, but with different expertise to coordinate and to get an idea on what is out there in terms of data, like disease-related data. So I think this is a very, very important step where still a lot of things needs to be done, or a lot of effort needs to be taken to get that rolling. And there are initiatives who basically have started that, and where people get together and get bioinformaticians, clinicians, and researchers together to do that.
And I think that's absolutely essential, which leads me to my next question regarding the cost action that you both spearheaded. Can you tell us a little bit more about the cost action to foster collaborations within the European community, and how did you decide to form this cost action alliance?
Well, basically, we've always been talking about that we need to include more people, actually, from a lot of different fields. I mean, the GPCR field is great. Everyone's highly connected. It's huge. But you start finding yourself in a position where your standard approaches just don't really answer your questions anymore. And we thought it'd be nice to have a platform for that, to have a name to it. And what is so nice about that cost action is basically something that first kind of worried us, But in the end, it's kind of cool because what happens is that people can just sign up. I mean, you need a bunch of people to start the network, but after that, it just basically flows free. And what's really necessary, besides those people having like already good tool sets and techniques, I think you need a lot of new people that are just totally naive and just propose or just do something. Right? Just not knowing that it can go wrong or expecting that it should be doing something great. They should just, do something out of the blue. And this kind of creativity is something that I often feel like is what we lose as we keep on doing this science and keep on digging deeper into our fields. And you need naive people coming from just everywhere. And that was basically the idea that we had behind it. And so we were sitting and looking up all the different things that need to be written down in order to meet EU standards for a grant proposal. And we got really lucky that we could also initially get quite a few people interested in joining in this endeavor, even though there was a bit of, I don't know, I think people were not sure if this could fly because there's already this really great GPCR cost action earnest that is so big and has all these great scientists. But luckily, we managed to convince also the EU committee that this is not going to be it. There's a lot more to do, and we need the people to do that.
That's phenomenal. Just to give an idea to our US listeners, the cost action is actually funding from the EU that allows to form foster collaborations and form this group. So you do get to apply for it, but then you get funding, and I think it renews every 4 to 5 years.
It does not really renew, so it's for 4 years, and then you have to show that you develop your network, and then you have to reapply, but with a different idea basically. So you need to show that you can develop, and you have a different perspective, a different angle. You have moved on. You have gotten stronger, and then you have the option of basically reapplying. Yeah,
which is
for another
4
years.
Which is, I think, pretty much phenomenal. I do ask this question from everyone on the podcast, and I usually give away the answer. But do you think GPCRs are still good drug targets? And if yes, which I hope the answer is yes, I'm pretty sure it will be. Why do you think that it is the case? Any of you, Simone?
Well, yes. Well, I was just thinking to say no, just to see what happens. But well, let's put it that way. Yes, they are good drug targets, and especially adhesion GPCRs. I still think they are. There are actually quite some GPCRs, or at least I know at least 1 or 2 where we are at the point where we have clinical trials going on. Speak electrophiles against pediatric asthma. So there is some medication on where they are in clinical trial. So which I think is a very good example for adhesion GPCRs, where we do know, well, we have talked about it a lot, that they do have a pharmacological potential. And even though we still don't really understand, as Ines said, we don't have modulators on hand yet, not that many. We still don't understand that much. We're already at the stage where we can actually design, with the little knowledge we have, we can design basically therapeutic. We can use them as therapeutic targets, where we, like I said, pediatric asthma is the example which I can give, where we have an adhesion GPCR, where we have a drug target which is in clinical trial at the moment, where people have moved to that point where they can do that, and I think there's so much more potential there. Yeah, I do think that they do have a lot of potential, and especially adhesion GPCRs with their large N-termini. I think there is a lot room for targets where we can they are difficult because they have so many functions, so we have to keep that in mind to draw apart all these functions, physiological functions, especially as they're expressed basically everywhere. You name a tissue, you name a process, and I name the adhesion GPCR, so we can make a game out of that. But they are great drug targets. I'm convinced of that.
Super. Ines?
Absolutely. No, I was just thinking we of course would love to see adhesion GPCRs as a drug target because there's just this whole new horizon of ways how you could tackle them. But what's also making this very, very difficult is that an adhesion GPCR is not an adhesion GPCR. You can have the same number, but you have so many different splice variants. They're tissue specific for all we know, just from the first studies. So you might not even know what are you targeting in the end, because the splice variant is decisive for, let's say, that the length of the N-terminus. That can decide what is the possible interaction partner. It definitely has an impact on the mechanoactivation. You might have splice variants that alter the 7-transmembrane domain. So even you find one compound, it might not really work there. So that brings us basically back to square one. We need to understand how many receptors are there. And 33 is just what feels like an arbitrary number, because what we're actually dealing with is probably hundreds, if that's even enough, right? And that's making it very, very difficult. But on the other hand side, once you know which ones are expressed in which tissue, in which certain condition of the tissue, right? It can change as a tissue starts getting fibrotic or whatever. And if you can even pin that down to an individual, you get a really good handle on personalized medicine, even though that phrase itself, the term personalized medicine, is something that I actually don't really like, to be honest, because it feels like so unfeasible. But I think once we have the tools to understand which receptor type is where, we might really have a chance of offering that.
Yeah,
so
I think
yeah.
I love the idea of, kind of mapping out the landscape and knowing which receptor, whether it's an adhesion receptor or not, which splice variants in what tissue, when and how does it modulate not only the physiological function, but also the pathophysiological function.
That would be the major aim. That would be fantastic if we could have that one day, where it would be a huge step apart to have like a huge map where you have expression, where you have modulation, where you have splice variants all in one go. That would be actually the main aim. Yeah.
That sounds like a great path forward, a great plan to get to that, mapping out that landscape. In my dream world, we would know everything about all GPCRs.
But then we wouldn't have anything to look into.
I'm pretty sure there would be something to look into. I think there's always something to learn about GPCRs in various contexts. All right, so let's move on to my next question, and it is about your advice that you would have for junior scientists who would want to contribute to the adhesion field or even just to the GPCR field. Ines?
Well, obviously, connect, collaborate. And one important advice that I wish I had before is always ask for more than you would think you're capable of. always try. You might be scared as crazy of taking up responsibility or going and presenting your data, whatnot. It doesn't matter how young or unexperienced you are. You just really got to do it. And that is really the point that I have to say this. I learned this from Simone, actually, because until I met Simone, I would have never thought that it's feasible to apply to give a talk at a conference or wanting to organize something, these kind of things. And she always said, yeah, of course. I mean, we should do that and try that. And I was like, never really thought of that, but yeah, why not? I mean, you might still not get it, but at least people know that you want to, and maybe at some point they remember, and then you get your chance. And it doesn't matter how scared you are before you go on that stage or you take up that responsibility for educating, teaching, organizing other things. You just do it.
Yeah, I love that. Thank you. I really love the fact that you just mentioned, just do it. Just because you're you're only a postdoc or you're only a PhD, that doesn't mean that you shouldn't go ahead and apply to give talks. That doesn't mean that you shouldn't promote yourself. Because when you do great science and you don't talk about that great science that you're doing and you don't exchange ideas, then it's almost in vain because you don't get to talk to other people. Simone, what are your advice? What's your advice?
Well, I can iterate a little bit about what Ines said, and I think one key is connect, network, get to know people also from completely different fields, not only from your field. Like, move out of your comfort zone, no matter how hard it is. Go to talk to physics, go to talk to like math people, and see what comes up. I'm always surprised when I randomly talk to people and try to explain what I'm doing, and then I get something back, and sometimes really cool ideas come up. And so I think that is really important to talk to people also from different fields, to like see what they have to say, and then to maybe get new ideas and especially new angles to your research questions, right? And this is basically also what the cost network is all about, like to connect. And then, yeah, you just have to try. I think what Ines said, this is very, very true. Don't be afraid. Don't be scared. Just jump and see what happens. And if you do good science, it will be worthwhile. And even if you just like fail to a certain extent, then it's science, right? I mean, things go wrong, experiments go wrong. Just try again. You learn from it. And in the end, it pays off. It's so satisfactory, right? To have, like, in the end, you have your data, you have your results, and you learned a tiny little bit about how receptors work. And I think this is especially for young people very, very important. And then obviously, basically, it has to be fun. So just try to see the fun in it. And science is fun, no matter how hard it is. I think that is the most important thing.
I love, to your point, I love the fact that you were talking about, failing and don't be afraid to fail. I think when I think about successes and failures, I think about successes as confidence builders. And I don't like the word failure. I call it lessons learned. You pretty much realized what you shouldn't be doing, which is also just as important as realizing what you need to do. Definitely. All right, super. So with this, I'd like to ask our guest attendees if they have any questions. Feel free to turn on your camera and ask questions to both our speakers. Let's just feel free. If not, I think no one has any questions, but if not, I want to thank you both for joining us today. I hope you had a great time listening to both our speakers. And with this, we're going to conclude our recording of the podcast episode. Simone, Ines, please hang back. Thank you so much for being here, and have a great afternoon. All right, let's wait another little 30 seconds if everyone has any questions. If not, this will be the end of our recording. Justine says thank you. All right. Thank you so much, and have a great afternoon, and have a great evening wherever you are in the world. It was a fun second recording of a live podcast. I hope you enjoyed it. All right. Thank you. Thank you for joining us for this Dr. GPCR podcast episode. I'd like to thank our guest, as well as our team members, Attila Forrest, Shivani Sajdev, Alexa Druan, and Ines Pinheiro. We look forward to seeing you live at the Dr. GPCR Summit in September. Don't forget to visit drgpcr.com/summit. Please subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us testimonials at drgpcr.com/testimonials. Another way to support us is to share your favorite Dr. GPCR program with your network and colleagues. Email us with any questions or suggestions at hello@drgpcr.com. Until next time, stay safe.